Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 22nd January 2021. As we are into the revision mode, we are revising the chapter today, chapter number 5, Principles of Inheritance and Variation. And this revision is for the third time in a row. First time we have given it revision notes. Second time I have trained you in assertion and reasoning. Third time I have given you test paper 1. Now I am giving one more paper, test paper 2, so that you will get familiarized with the type of questions as well as the way you have to present your answers to the examiner. So here we go. Question 1. Two alleles of a gene pair are located on homologous site on A. Sex chromosomes, B. Autosomal chromosomes, C. Homologous chromosomes, D. Heterologous. I think homologous. The answer should be homologous chromosomes. Alleles present on place, locus. Which of the following is not a deviation from Mendelism? Okay, MEN, Mendelism. A. Multiple alleles, ABO blood grouping, dihybrid cross, and incomplete dominance. I think A, B, D are all deviations, and only Mendel's is dihybrid cross to be followed. Third, in our society, females are blamed for producing female children due to A. Scientific reason, B. False notation, C. Production of same kind of gametes, D. Biological concepts. I think in our society females are blamed. False notation, yes, wrong concept. Next, question number 4, assertion. Chromosomes and genes both occur in pairs. Reason. The two alleles of gene pair are located on homologous sites on homologous chromosomes. Reason is not related to assertion. Assertion is correct, but reason is incorrect. Assertion and reasoning both are correct. Yes, both are correct. Assertion and reason both are incorrect. So answer should be C. Next, question number 5. When two genes present on different loci produce the same effect when present alone, but interact to form a new trait. Trait means character. When present together are called polymeric genes, duplicate genes, complementarity. Yes, they complement each other. Complementary genes, supplementary genes. So answer should be C. Question number 6. What kind of test will you perform to find out whether the given plant is homozygous dominant or heterozygous? Question 7. A garden pea plant produced axial white flowers. Axial white flowers. Another of the same species produced terminal violet flowers. Identify the dominant traits. Okay. Garden pea plant it is. Right. Question number 8. Go for the cross. Question number 8. Which one of the following diseases could be avoided in the progeny by analyzing the pedigree of the parents? Down syndrome, phenylketonuria, and poliomyelitis. Which one of the following diseases could be avoided in the progeny? Yes. Okay, next. A man, question number 9. A man of A blood group marries a woman of AB blood group. Which type of progeny would indicate that man is heterozygous A? Okay, so you have to go for the genotype, and cross verification can be done. Question number 10. The following table shows the genotypes for ABO blood grouping and their phenotypes. Fill in the gaps left in the table. Recollect from your memory when I was doing you regular video tutorials. In that video I had given the same type of table. Okay, genotype, blood group, and this phenotype as well as this. So wherever blanks are there, like IA is A, homozygous, then it has to be even IA I0, for example. Okay, that also becomes A. Like that, we can fill it up. Question number 11. Write the sex chromosomal complement of the following. So animal name he has given and sex chromosomes he has given. Drosophila, grasshopper, fowl, Homo sapiens. So you have to write the sex chromosomal complement for the following. Okay. Question number 12. A dihybrid heterozygous round yellow seeded garden pea was crossed with a double recessive plant. What type of cross is this? Work out the genotype and phenotype of the progeny. What principle of Mendel is illustrated through the result of this cross? Question number 13. This is very important. Case study based questions I said. This is for 4 plus 4, 8 marks for your batch. So take care of such questions. Question number 13. The biology teacher asked the students to verify the experiment on transformation principle in bacteria to establish DNA as genetic material. The class was divided into 2 groups. The teacher asked them to submit the reports. Group 2 did not use the mouse and did not repeat Griffith's experiment. The teacher praised them. What values did the group 2 exhibit? Question 2. Which experiment did they perform? Explain in brief. Okay. The thing is they have to establish the DNA is the genetic material. Okay. So the second group wanted to avoid the animals. They don't want to kill the animals. That is what. Question number 14. In pea plant, a smooth seed coat is dominant over wrinkled seed coat. So seed coat smooth. And what will be the expected ratio of phenotypes of the offspring in a cross between heterozygous smooth versus heterozygous smooth? Heterozygous smooth versus homozygous wrinkled. Heterozygous smooth versus homozygous smooth. So first arrive at the genotype. Based on the genotype, you can go for the cross. Question number 15. A woman with blood group O married a man with AB group. Show the possible blood groups of the progeny. So write the genotype. You can write list the alleles involved in this inheritance. Okay. Now take a paper, start writing the questions, and you may take say 45 minutes, and then compare your answers with the answers that I am giving now. So these are the answers. Question 1C. Homologous chromosome. 2 alleles of a gene pair are located on homologous site on homologous chromosomes. That's why we are calling homo means similar. During gamete formation, homologous chromosomes separate from each other, carrying one set of allelomorphic genes. Question 2. Dihybrid cross. The events that do not follow Mendel's laws of inheritance are called deviations from Mendelism. Incomplete dominance, ABO blood grouping, multiple alleles are deviations from Mendelism. So dihybrid is the only one that is there. Question 3b. False notation. In our society, females are blamed for producing female children due to false notation, although male sperm decide the sex of the baby during fertilization. Question 4. C. Assertion and reasoning both are correct. In diploid cells, both chromosomes and genes occur in pairs. 2 alleles of gene pair are located on homologous sites on homologous chromosomes. Question number 5. C. Complementary genes. Complementary genes are one of the 2 or more genes that, when present together, produce effects qualitatively distinct from the separate effect of any one of them. So complementarity genes, like I gave the example of the 2 Indian pace bowlers bowling in a test match, like Jogal Srinath and Venkatesh Prasad. Many times the commentators used to tell complement each other. They complement, like Jogal Srinath bowls an in-swinger, Venkatesh Prasad bowls an out-swinger, like that, and vice versa. So they complement each other. Like that, complementary genes, they complement each other for a phenotype. Question number 6. Test cross will be performed where the given plant is crossed with a recessive plant. So it is the test cross. So where given plant is crossed with a recessive plant. Next, which is dominant, axial and violet. It is based on the numbers. How do you arrive at the dominant character or recessive character? Based on the numbers that are available. Which numbers exhibit this particular phenotype? Maximum numbers exhibit in a population, that is the dominant character. Then phenylketonuria, we can drop it out. Next, question number 9. A man of A blood group marries a woman of AB blood group. Then the progeny of B blood group can indicate that man is heterozygous. Okay, heterozygous. That means it has to be IA-IO. So you just keep this IA genotype I0. Another one is of course homozygous IA. And since it is versus IA-IB. Okay, so if this is the combination IA, the phenotype is A. If this is the combination, phenotype is to be B. And so as this with this, same. And IA-IB, then you got the possibility of AB also. But in case of this, since we are arriving at the B blood group, this indicates that it is not homozygous, it is heterozygous. So if the male parent is homozygous for A blood group, then only possibility of progeny will be showing either A blood group or AB blood group as we listed. If the male parent is heterozygous for A blood group, then the possibility of progeny will be showing A blood group, AB blood group and B blood group. So this is easy way, right? Genotype and go for the cross. Then you can easily arrive at the conclusion. Next, thus the distinction would be made only on the basis of progeny showing B blood group. Next, based on again multiple alleles blood group, I said IA, it is pure homozygous, phenotype is A. IA, I0 or IO we say A. IB, of course straight away B. IB, I0 again B. IA, IB, it is AB group. And I0, both are zeros, then it is O blood group. Question number 11. XY, X0, of course in the case of birds it is ZW and XX. These are the chromosomes related to the sex phenotypic. And next, question number 12, it is of course test cross. And the best way to explain this is to put the checkerboard or Punnett square. So yellow round seeds and green wrinkled seeds. So you need to take up the capital ones, yellow round, capital Y. But here he is taking it capital Y, capital R, small r, because it is homozygous. And homozygous is crossed with always the recessive ones, small y, small r. Thus we get the ratio 1:1:1:1. Yellow round, yellow wrinkled, green round, green wrinkled. So, and this shows that there is separation of the genes. And that is why principle of segregation is the involvement here. So test cross is always with heterozygous plant crossing with the recessive one, okay, recessive homozygous one. Question number 13. Scientific attitude, awareness and love for animals and respect towards government policies. That's how the batch of students saved the mice. The students repeated the experiment performed by Oswald Avre, Colin MacLeod and Maclean McCarthy, who worked to determine a biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals like proteins, DNA, RNA from the heat-killed S cells to see which ones could transform live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria, rough bacteria, to become transformed. They also discovered that protein digesting enzymes, proteases, RNA digesting RNAs did not affect transformation. So, the transforming substance was not a protein nor RNA. Digestion with DNAs did inhibit transformation, suggesting that DNA is the only reason for transformation. So, that's how we need to appreciate. And Draw the diagram. S phase and R phase, S strain, smooth and rough strain. This is how we can prove that DNA is the only genetic material. Next, 14th, smooth seed coat, dominant, wrinkled seed, smallest, recessive. Remember, I gave a suggestion to you that whenever you are taking genotype, the letter has to be like if you are taking the smooth seed coat, the spelling is smooth and first letter is S, you need to take capital S because it is dominant. When you have taken this capital S, even though wrinkled spelling starts with W, though you should not take small w, you need to take small s only so that you will not have confusion. So heterozygous smooth, heterozygous smooth. So capital S, small s, capital S, small s, because heterozygous and it is smooth, S is dominant. So crisscross, you get this capital S, capital S, capital S, small s, capital S, small s, and smallest, smallest. 1:2:1. That should be the F1 progeny. So as you are seeing, 3 smooth, 1 wrinkled. So in 3 is heterozygous, 1 is homozygous. Second type, parents capital S, small s, and smallest, smallest, recessive. Then it is phenotype 2 smooth, 2 wrinkled, because the genetic combination is capital S, smallest, 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 capital S, smallest, and smallest, smallest. And next category, heterozygous smooth versus homozygous smooth. Heterozygous capital S, smallest, and homozygous smooth capital S, capital S. Now, what is the resultant of the F1 generation? All are smooth. Like capital S, small s versus capital S results in formation of 4 gametes. Capital S, capital S, capital S, capital S, capital S, small s, capital S, small s. And in all this you see the dominant gene capital S, capital S, capital S, capital S. It has to be 1:0. So the gametes that are formed in question number 15, IO from mother, because mother belongs to O blood group, father AB blood group. So since it is IO, the gametes will be having genes I0, and in the case of father it is IA, IB. So crossing I0 versus IA. So A group heterozygous, I0 versus IB, B group heterozygous. So possible groups A and B. Alleles IA, IB, IO. Hence the 3 alleles are involved in that. So in this chapter particularly you can score out of what? Because it is just like mathematics. You get that Punnett square correctly, you write the parental genotype correctly, and segregation of alleles right correctly, you get out marks. So this chapter don't neglect. You will get full marks if you practice this monohybrid, dihybrid and variations of the crosses. What is important is you should correct